Okay.
And Natalie, like last week, I'll introduce you, and then you can go ahead and get started.
Okay.
Okay. Okay. So it's my pleasure to introduce Natalie Justice, and her presentation is from an article entitled "Evidence for a Trophic Cascade on Rocky Mountain Reefs Following Sea Star Mass Mortality in British Columbia."
Okay, hi everyone. Thank you all for coming today, and thank you for that introduction, Dr. Appel. I will be presenting a little summary of a report about the recent sea star mass mortality that is occurring in Howe Sound, which is located in the British Columbia, as you can see on this map to the left. By a show of reactions, has anyone ever heard about this topic before? Okay, I don't think I saw many, but so I'm not sure if anybody has any background knowledge, but for those of you who don't know, the sea star wasting disease, okay, the sea star wasting disease is, as you can see in the photo progressions to the right, is basically a disease that many echinoderms have been getting in the last probably about 2 decades from what we've been recording. What happens is the sea star begins to turn a little bit white, and slowly their limbs disintegrate into nothing. While research is still being conducted for the cause of the disease, it is ultimately unknown as of now. However, many scientists have concluded that it can most likely be drawn to ocean acidification, drought, pollution, and bacteria increases with nutrients from eutrophication. And why is this important, you may be asking, is because sea stars are a keystone species, which means that they're drivers of ecological ecosystems. So as a predator, they, to many other species that can be deemed harmful, such as sea urchins and barnacles, which I'll go further into in the upcoming slides, they are responsible for keeping the biodiversity of intertidal zones well balanced. Okay, so for the objectives of the experiment, they were to observe changes in the community before and after the sea star mortality to prove evidence of a trophic cascade. And for those of you that aren't like quite positive what a trophic cascade is, it's basically an ecological phenomenon where you're either adding or in this case removing top predators, which causes really fast changes in relative populations that are surrounding them in terms of predator and prey. So in other terms, like the food chain undergoes drastic changes in structure and nutrient cycling. And yeah. So for how they gathered their information, they did subtitle surveys through the use of transects, which I'm not sure if everybody here has taken NR 306, but for ENVM we do. And you actually do something very similar to what these scientists did in Pismo Beach, where you basically use transects to count individuals. So out of 17 sites, they had 4 transects in each, which were 25 meters long and 4 meters wide. And within these transects, they counted individuals by species over about a 4-year period. And on top of that, there were also citizen contributed reef surveys, which are basically citizen scientists and trained scuba divers that reported their findings when they were out and about doing whatever they were doing. So here is a chart for some of their results that they found. And basically the main thing that I want you guys to look at here is the sunflower stars and the green urchins, which are the top 2, not the invertebrates. And as you can see, so the mean densities before and after go from 6.4 to 0.9, and then the green urchins go from 18.3 to 77.2, which is a huge hop in population. So as green urchins increase in population, it has a huge effect on kelp. And kelp is a main food source for sea urchins. And basically, when sea urchins, how they kill kelp forests is they eat the root of the kelp. So that keeps the kelp anchored to the bottom of the ocean. And kelp forests are really important because they provide food and shelter for thousands of species, and they harbor a great variety and diversity of plants and animals than probably any other ocean community. And as you can see in this graph here, you can see the abundance scores for sunflower stars decreasing as the epidemic was first observed, and how the green urchin populations increase. So in conclusion, the 90% decline of sea stars has a trickle-down effect for the entire community because sea stars are predators to species such as sea urchins and barnacles. They're responsible for maintaining the biodiversity, because without sea stars preying on these other two main species, they will take over the zone and make it difficult for other species to exist spatially, and they have to fight for food and nutrients. So overall, further research is needed to ultimately understand the cause of the wasting disease, but this is one of the largest scaled subtidal trophic cascades that we have yet to record. And I think I went really fast, so I have time for questions.
Yeah, we do have time for one question probably. Any questions?
I have a question, Natalie. What made you want to choose this topic?
I learned about it when I was in my NR 306 class, because our teacher was like, if you find any sea stars, like, let me know, because you can, even it happens everywhere in the world, it's not just occurring in the British Columbia. So it's happening everywhere, and as a citizen, you can like go onto this website and record if you see one. And I just wanted to learn more about it. So, and I thought it was kind of a fun topic.
Anything else? All right, good job. Yeah. So our next speaker is Siobhan. So Siobhan, you should be able to share your screen.
View, present, cool. Can people see this?
Yep. All right, so I will introduce you. All right, so it's my pleasure to introduce Siobhan Kassem, and she is going to present a talk entitled Ecotoxicological Responses of a Reef Calcifer Exposed to Copper Acidification and Warming.
Thank you, yes. So I chose this because I grew up next to an ocean, and I've always been fascinated with the environment there, and I've gone to many coral reefs, so I wanted to learn more about it and what's affecting them. So yes. It's not letting me change. Oh, there it goes. So I'm going to be going over the objectives, background, materials and methods, discussion, and conclusions. So the objectives are to investigate the effects of ocean acidification and warming, in addition to copper co-exposure on mortality, bleaching, respiration, and growth of corals. The next objective is to evaluate the general stress using the integrated biomarker response approach. And this IBR monitors the effects and grade of pollution and assesses general health using multiple biological endpoints. So for some background, since the pre-industrial era, sea surface temperature has risen nearly 1 degree Celsius, and the pH has dropped by 0.1. which doesn't seem like a lot, but on the pH scale, that can have detrimental effects. And as these temperatures and pH change, as the CO2 emissions rise, the marine environment is especially affected. Corals are calcifying organisms that help build the construction of the reef, so acidification is very destructive. And metal contamination is on a local scale, which can come from agricultural runoff, domestic sewage, and some paints. And excessive concentrations of copper can be toxic and inhibit calcification in these coral reefs. So the materials and methods, there were 4 tanks set up in this experiment. The first tank was an ambient tank, which mirrored the natural environment. The second tank showed ocean acidification as the pH was reduced by 0.3. The third tank showed the warming, where the temperature was raised by 3 degrees Celsius. And the fourth tank showed both ocean acidification and warming, as it had both the pH reduced and the temperature raised. There was also a second set of 4 tanks, which had the exact same things as these tanks, but also had the copper inclusions to test whether the copper plus the reduction of pH and the warming had other effects. So for the materials and methods, coral was collected in a marine conservation park in northeast Brazil, and they were placed in a lab, and then they were placed in the cages in the tanks with the local sea water and with natural sunlight cycles, and there was mesh to limit the movement. And these equipments, the first one, the stereo microscope, was used to assess the amount of dead individuals in the tank, and that number was divided by the total amount in the tank as well. This was also used to look at the amount of bleached coral. The micro respirator was used to look at the respiration rates by changing oxygen concentrations during a 15-minute dark phase on this machine. And then the BioDrop light was used to look at the biological parameters, and chlorophyll was extracted, and a total protein concentrations were looked at with this device. So this is the first graph that's showing the mortality of the corals, and you can see that it is divided into 2 parts. On the left-hand side, it is the 4 tanks, which are indicated by the different colors, and it's without the coral. And on the right-hand side, without the copper. And then on the right-hand side, it's the 4 tanks with the copper additions. And then on the y-axis, you can see the percent mortality. And as you see, the red box is the highest, showing the highest mortality with warming effects of the tank. And then the next one closest would be the ocean acidification and warming in addition to the copper. This graph is showing the bleaching of the coral, and you can see the 4 tanks on the right are much higher than on the left, showing that the copper additions to the tanks had drastic effects on the coral and the bleaching frequency. This is the size and growth graph showing the size of the coral, and the gray box is the ambient tanks, and as those 2 boxes with and without copper are relatively the same, we can see that ocean acidification, warming, and both of those combined have very reduced growth and size compared to the natural environments of the coral. This is the respiration rate graph, where it shows the obvious purple tank on the right with the copper had a much higher respiration rates due to ocean acidification and warming in the tanks compared to the ambient tank on the left-hand side with no other additions. This is the calcification-related enzymes graph, and you can see the box on the left in gray, that is the ambient tank, and it is much higher compared to everything else, showing that all the other boxes, whether it be ocean acidification or warming or both, had very negative impacts on calcification. And this is important because again, the corals are building their structure and their environment out of their enzymes. And if they don't have these calcification enzymes, they can't build their structure and they could die. And this is the IBR index graph. And again, this index shows the stress of coral. And we can see that the ambient box on the leftmost side is the lowest. And the main takeaway from this graph is just seeing that every other tank and box is much higher on the y-axis, showing the amount of stress that is present in these tanks due to either the reduced pH or the increased temperature. And for some conclusions, as we've seen it in the previous graphs, the co-exposure to ocean acidification and warming and copper resulted in the highest mortality and bleaching, along with respiration rates, reduced growth, and calcification related enzymes. And these all drove the high IBR stress index in indicating health impairment of coral under these multiple stressor conditions. The integrated biomarker analysis evaluated that the copper contamination reduced the tolerance of coral to climate change. And this is a picture of coral in American Samoa, and it's showing a year difference in the same coral. And this is just showing on the left, a live and healthy coral, where on the right is a dead and dying coral. And a take-home message that I want to leave you guys with is the next time you're out at the beach and see coral reefs, take a chance to look at them and appreciate them as we can, because as the CO2 emissions continue to rise, it's unsure how long these corals will last in those types of conditions. So we should see these corals while we can. And thank you. Any questions?
Yeah, time for one question. Okay, thank you very much, Vaughn. And we'll move on to our next speaker, which is Justin Lack. Okay, so it's my pleasure to introduce Justin Lack, and he's going to present on an article that was entitled Spring Flow, Bacterial Contamination and Water Resources in Rural Regions. You're muted, Justin.
Can you hear me now? Can you hear me, Dr. Appel?
Yep, go ahead.
Okay, perfect. Yeah, thank you for that introduction. And before I begin my presentation, I just want to start out with a quick question to the audience. And using your poll with a thumbs up emoji, how many of you have turned on your water faucet today? All right, I see quite a few. And then a second question, how many of you have thought whether or not that water was safe to drink? All right, much fewer. And in the context of the study, it's important to note that millions of people worldwide do not have access to clean water and sanitation. And this article is really important to me because over the past summer, I traveled to Haiti and I saw firsthand some of the challenges that these people face in regard to water access. And I'm excited to share with you today what the article is all about. And so let's get right into it. So today I'll be talking about the study and what they found. First, we'll go into the context of the study, why it was important. Then we'll go into the objectives, what were the researchers trying to understand. Then we'll go into the methods, how did they conduct the research, what materials did they use. We will find out about their results, talk about it through their discussions, and finally end with a conclusion. And so a bit of background. So the pair of researchers that conducted this study were two professors from, and they were teaching environmental geology, and they traveled to Biarritz, Haiti. And if you're not familiar, Haiti is located on the southern tip, or off in the Caribbean Ocean, off of Florida and Cuba. And this is a really significant site because Haiti experiences it's challenging for water access for people. In fact, about 4.3 million people do not have access to clean water in Haiti. And according to the World Health Organization, 1 out of every 3 children in Haiti pass away before the age of 5 due to waterborne illnesses. And so the first objective of this study is to identify if the groundwater springs in Biarritz, Haiti is safe to drink. And the second objective of the study is to understand if this water quality is not up to par, maybe like why is it bad? And so on the right, I have a diagram of what groundwater is. And so you have a rainfall event. As rain infiltrates through the soil, it collects as groundwater, and then it emerges as a spring. And on the left is kind of a hint that I want you to think about for why might groundwater be have poor groundwater quality. And so for every spring that the researchers went to, they had 2 different testing. Oh, and before I say that, there was 26 springs that they sampled within the town, and 1 sample they sent to the laboratory for more rigorous testing of water quality and the coliform and bacteria that was present. And in the right-hand picture is Coliskin Easy Gel kits, and they use this as a field measure because it was quick and inexpensive and an easy way to measure if there is bacteria present or not. And you can see the blue and pink dots represent if there is bacteria present. And so this is a really significant graph. And to understand how I'll break it down. Here on the x-axis, we have the type of bacteria analysis method used. And so for example, here's the hospital bacteria analysis. This is the laboratory in the hospital. And on the right side column, we have the Coliskin Easy Gel kit. And this was again in the field. And the y-axis measures the percent total coliform. And so in summary, 71% of the samples they did, according to the laboratory, measured that 71% were unsafe to drink, according to WHO standards. And regardless of the sample that they used, this is a majority of the drinking water in the town. And a second research question they were asking is if spring capping improves water quality. And this is something I had trouble understanding, so I want to help explain it to you. The spring capping refers to the man-made structure they make around springs, and this helps in theory to improve water quality because it reduces surface contamination. And the bottom picture shows a spring that is not capped. And this graph reveals that regardless of the type of spring, they still have the majority of water is still unsafe to drink according to WHO standards. And there's a large discrepancy between the numbers. As you can see, the capped spring has 66% unsafe water, whereas the uncapped is 81%. But this gives us indication that maybe that contamination of the water quality happens before, kind of in the groundwater. That leads us into our second objective, which is why is that groundwater spring quality poor? And there's 3 factors that we're going to discuss today. The very first is shallow karst aquifers. And here's an image from the study, and it's a geologic map. And if you look at the colors, the pink and the green teal represent carbonate aquifer, and an aquifer is the medium or the rock unit or the soil that holds the water. And this is significant because carbonate, it can dissolve with the water and form large cavities and fissures within the rock. And the diagram on the left shows just that. And as water infiltrates down, it can easily transmit through these pores and emerge through the spring. And one result of this is that the natural filtration process of aquifers is diminished because it can transmit so rapidly. And the second part is deforestation. And I was able to take these pictures when I was in Haiti this summer. And on the right is actually the Dominican Republic. And it shows kind of the natural vegetation that is native to the area. And on the left is a picture of Haiti and the deforestation that is in place there. And this is really significant, but only 2% of Haiti's trees remain. The rest is deforested. And this impacts the water quality because as rain infiltrates down, there's less filtration because the soil has already been eroded away. And the third factor is poor sanitation. And on the right, I want you to imagine that you're at this marketplace. And this is atop a ridge crest in Haiti. And imagine being in this village and you have to go to the bathroom. I just want to point out that there is no public restroom. There's no sewer system. There's no drainage. And so the diagram on the left shows that as animal and human feces gets washed from the rains, it travels through the soil into the groundwater spring, which it can easily transmit into the springs that people use to collect water. And so today we have learned that there's quantitative evidence that the majority of water in Varetz, Haiti is unsafe to drink. We have learned that while capped springs do provide slightly better water quality, the main issue is what happens before that groundwater emerges as a spring. We've learned that the main causes within the scope of the article, the main causes of poor water quality is deforestation, shallow karst aquifers, and poor sanitation. And the researchers agree that that much needed research needs to be done. It was one of the first of the kind research articles in Haiti specifically on groundwater, and so much needed research needs to be done. And this leads to our conclusion. And so the next time you turn on your water faucet, I want you to remember the people of Haiti and the challenges that they face, but not to leave it as a problem, but to go back to your institution, Cal Poly, and to really be inspired about how your learning can solve critical global humanitarian needs. Thank you.
Questions for Justin? I have one question, Justin. Were they able to come up with a way to compare how much of the poor water quality or the coliform contaminated water was associated with animal versus human waste?
That's a really good question, to differentiate between animal waste or human waste. And they did not specify in the article, and that's a really good research question for another research task to go about.
Okay, we will move on to our next speaker, and our next speaker is Olivia McKay. So Olivia, you can share your screen.
Can you see it?
Yes.
Okay.
Yeah, so I'll introduce you. So today, Olivia McKay is going to present on an article that was entitled "The Role of Geothermal Resources in Sustainable Power System Planning in Iceland."
Okay, so a fun story about this picture. I actually took it myself when I was visiting Iceland and studying at the Iceland School of Energy. I was one of 4 people out of 56 people on our ship that didn't throw up from being seasick, so I'm pretty proud of myself for that. But in all seriousness, I think what distracted me from the seasickness was the beauty of the country. I mean, it is just so amazing there and so beautiful, and I was so impressed with how the citizens there treat the environment. And so that's what inspired me to learn more about the renewable energy there. So today I'm going to give a little bit of background on renewable energy in Iceland, and then go over the objectives of the experiment, the materials and methods, and then finally get into the results and discussion, and then end with a summary and conclusion. So Iceland is a really small country. There's only 360,000 people about. But what's amazing is 100% of that 360,000 have access to electricity. They're able to do this because the electricity costs are so inexpensive, and that is due to the access to renewable energy. So 98% in 2017, 98% of renewable energy came from indigenous, 98% of the electricity came from renewable energy usages. And so that's why they're able to keep the costs down. But there was 81% overall of the demand for electricity was, not electricity, of energy was from renewables. So that remaining percent is still fossil fuels. They made this experiment to lessen the use of the fossil fuels. And so they came up with a goal of sustainability. So that was to decrease fossil fuel usages and lower the impact that fossil fuels have on the environment, but while still keeping in mind the economy, because this was written in 2019 and we do live in a world where fossil fuels are essential. And then keeping in mind that dealing with renewable energy can be tricky because it might take a little bit of time to recharge. So you want to make sure that the consumption rate is less than the recharge rate. And then this is an image of the fossil fuel usage in Iceland, and majority of it is for fishing, ocean vessels, and then transportation, which are two of the main industries in Iceland. So they focused on that. The objectives are to understand the interaction between geothermal and electrical demands, grasp the relevance of renewable energy for Icelandic peoples, create an energy and system transport model, and then they want to connect this model to a geothermal resource dynamics model. And so it's connecting the need and the demand to the supply, so adding a little bit of economics into it. So the materials and methods that they used were connecting these two models and running 16 different scenarios through them. And then they were all based off of assumptions and projections from the year 2020 to 2050. The first model is called UNICID, and it's Iceland's energy and transport system model. So this is for the demand and the energy costs, and then they connected that to a geothermal resource dynamics model, and then that is dealing with what the wells and the geothermal power plants can provide to meet that demand. And then this picture below is an example of what these power plants look like. And so, believe it or not, this is the simplified version of the model. It's still a little bit confusing, but when you just look at it color-wise, the pinkish red color is the UNICID model, which is the demand, while the green is the geothermal resource model, and then that is the supply. So when they were making this, they considered a lot of different factors like air quality, water quality, the stock supply, and then projected stock supply up to 2050. And so with this, they were able to come to a conclusion, and they actually looked at it with two different scenarios. So including the feedback, which is the Y that you can see here, and then disregarding the feedback, which is N. And then every time that the feedback was disregarded, it led to a greater share of electricity as well as GDP growth, which sounds ideal because, you know, the economy is boosting while there's more electricity available. But we all know that we can be greedy as humans and we shouldn't be depleting our resources. So in summary, it is essential to consider all of these factors when you're assessing energy pathways. And I think it's something that we should do in the US a bit more. but increased GDP growth rates and electricity demand leads to a decline in what's available. And so again, while that sounds nice, it doesn't always end nicely, and it will eventually negatively influence electricity availability and affordability over time. So when I stated that 100% have access to electricity, that would definitely go down because it would become more expensive, and we wouldn't be able to enjoy nice hot springs like these, and which is something that I definitely did over my time. So does anyone have any questions? Thank you.
Questions for Olivia? Olivia, I have one. So did you say at the very beginning that there's 98% of the energy that they use there is from renewable sources?
Yeah, well, 98% of electricity and then 81% of energy in general. But the electricity is the 98.
And
I know, it's amazing.
I can't remember what you said, but what is their, so their main source is geothermal, right?
Geothermal and then a lot of hydropower.
That's amazing.
Yeah, I got to go to a lot of the plants there, and it's kind of crazy. And then for, oh, I don't know how to go back, but the Blue Lagoon that everyone goes to, it's actually just made of wastewater from the geothermal plants. So kind of crazy.
Okay, well, thank you very much, Olivia. So our next speaker is Max Niemeyer.
How do you stop sharing your screen?
Let's see. I think at the very bottom of your screen. Yeah, there you go. You did it. And just a reminder to all of you in the audience, if you can, please show your video so it looks like the speaker is presenting to an audience. And remember that my expectation is that you will be evaluating everybody and that you will post their peer reviews to the appropriate OneDrive folder. Next. Max always sees a dark screen.
Hello, hello.
Yeah, we can hear you just fine.
Okay. Yeah, I'm having some issues. My computer's a little on the older side, so bear with me.
Yeah, if you want, we could. Oh, there you go.
Oh, is it up?
Yeah, it's up.
Okay. Let me get it into full screen. Hopefully, if it'll let me. Come on. Okay. I think that worked.
Yeah, it looks like it's full screen.
Okay.
So I'll go ahead and introduce you. It's my pleasure to introduce Max Niemeyer and his presentation entitled Predicting Soil Organic Carbon Content Using Hyperspectral Remote Sensing in a Degraded Mountain Landscape in the Lesotho.
Okay, well, thanks for that introduction. Just looking at the title of the journal, it can be a bit daunting, but I could break it down. It's essentially just a study of an unconventional way to measure the soil organic carbon, and it just happens to be in a degraded mountain landscape in the country of Lesotho. Okay. Okay, well, I'm having some technical difficulties.
Or do you want to?
Can I be skipped and come back around?
Yeah, we'll do that.
Okay,
Okay.
I'm sorry about that.
That's okay.
Jeez.
So we, Jason, let me Jason, share screen. Okay. Jason, you should be able to share your screen, and Max, you can stop. There you go. Jason, you good? Okay.
All right, can you hear me now?
Okay.
Can you see that? All right. All right.
So, all right, it's my pleasure to introduce Jason, how do I pronounce your last name again?
Pitsilidis.
Pitsilidis, and his title for his presentation is entitled "Can Plants Grow on Mars and the Moon? A Experiment on Mars and Moon Soil Simulants."
Thank you, Dr. Appel, for that introduction. So today we're going to be talking about the future. As some of you may know, the US and other countries have put together a plan to go to Mars and the moon again. And so if some of you have seen the movie The Martian, it plays around with the idea of growing plants on Mars. But how do you do that? And how do you create a sustainable colony on a planet? You need to grow plants and you need to grow crops to grow food. And this experiment done by researchers in the Netherlands does that by growing plants on Mars and moon soil simulants. So a little overview of what we're going to be talking about today. First, we're going to be going over background of the experiment, the objectives of the experiment, what kind of materials that they used in the experiment, the findings, and finally, the conclusions and further research. So let's go into a little bit of background. How do you get a soil simulant, and what is a soil simulant? Well, we know that we have rovers on Mars and that we've sent humans to the moon. And those humans brought back soil samples, which the scientists have been able to analyze and recreate soil simulants which have the almost identical chemical compositions. And they've done that the same way with Mars soil. They've done that by remote sensing from the Martian rovers. The other key component of this experiment is looking at the nitrogen cycle. If some of you have taken plant health and soil nutrition, and you might know that nitrogen is one of the key components of soil in which it gives health to the plants, and it's very essential. It's an essential nutrient. And how do you find nitrogen on the moon and Mars? Well, there hasn't been evidence yet, but scientists speculate that nitrogen can be produced from active atmospheric conditions such as lightning on Mars. And this is why they've chosen to use nitrogen-fixing plants in the experiment to see if on Mars, if they can fix nitrogen into the soil to create a more nutrient-rich soil for growing crops. So what were the objectives of the experiment? Scientists planted species of 3 groups in artificial Mars and lunar regolith soils. And they did this to determine if it was feasible to grow plants outside of Earth and to create colonies on Mars and on the moon. So what were the materials? They used 3 soils. One was the earth soil from the Rhine River, specifically 10 centimeters below the surface. They did this in order to get a low nutrient soil so that it could be comparable to the lunar and Martian regolith simulant. They also bought the lunar and Martian regolith simulant from JPL Labs in Pasadena. Now they analyzed these soils and have found that all 3 of them have really high pHs, that the lunar and Martian simulant have traces of nitrates, and the Rhine River soil and the Martian regolith simulant have traces of carbon. And these all affect how plants react when planted in them and growing. So what kind of plants were used? Well, we have three groups: crops, nitrogen fixers, and naturally occurring plants. And here you can see that we have tomatoes, carrots, and you can probably recognize from the plants, or the naturally occurring ones, field mustard and stinging nettle. Now, if you've taken plants in plant health and soil nutrition, you might know that we use nitrogen fixing plants as cover crops in order to fix nitrogen into the soil to help the crops grow. So that's why we have the nitrogen fixing group. So what were the methods used? This is a pretty standard experiment in which they created replicates of the soil and plants. So they have about 80 per soil and plant, and created about 800 of these pots. And on the left, you can see a map of the greenhouse in which they have a random assortment of the plants, so that there isn't any way that there could be a favorable way that these plants are growing in the greenhouse. So on to the findings. What happened in the experiment? Our first graph shows percent germinated. And here you can see that the crops are in the highest percent germinating. And this is very surprising. As you can probably see on the bottom, we have the legend, which shows the earth soil, moon soil, and Martian soil. And we all see here on the trend that the Martian soil simulant is highest. And we see that the lunar soil is the lowest, and Earth is around in between those two. And of course, you can see that some plants were able to germinate higher than other plants, especially in the occurring naturally, where there's A. montana versus in the same group, the F. rubra. So on to the next finding, we have total biomass. Now, the total biomass is basically how much of the plant has been produced with this. And again, you see the trend where there is a higher biomass with the Martian soil, and you see lower in the moon soil, and again, a medium with the earth soil here. And again, we see with percent leaves how there is a higher percentage with the Martian soil here as then with the lunar soil and the earth soil, but the crops have a higher percentage of leaves with the Martian soil than any of the other plants. So on to the percent flowers, and this is a very interesting case where most of the plants didn't flower, but the ones that did, most of them were in the Martian soil. And this is a very interesting result, as it's not very common to have this, the Martian soil is not very nutritious and doesn't have any nutrients as the earth soil does. So the experiment lasted for 50 days. And after 50 days, you can see here on the graph that most of the plants that survived were in the earth and Martian soil. The ones that didn't really survive were in the lunar soil. This is because, again, like I said, the lunar soil did not have as much carbon as a nutrient. So what did the researchers conclude? We concluded that crops did better in the Martian regolith. And this is, again, because the earth soil is a comparable one where there isn't as many nutrients, but again, it's not the kind of soil that you find in farmland where they've inputted nutrients as well. We also found that Martian soil had a higher biomass, and this is good because you want a higher biomass in that when you go to a Martian colony, you want to have higher yields and more food for the colonists. They also concluded that further research was needed to kind of look at the physical aspects of these soils and how they affect plants as well. On this slide, I have a picture of a Chinese lander that landed on the moon recently with a cotton plant, and it only survived for a couple of days, but the cotton plant was able to sprout. And here you can see that that just means that further research is being put into having plants be incorporated on colonies on Mars and the moon. So if there's any other questions, please let me know. And thank you for watching.
Time for one quick question.
Yep.
Jason, I'm curious about the quantity of nitrogen in the atmosphere of the moon. Like on Earth, it's like 78%. Do you know what it is on the moon's atmosphere?
Well, since the moon doesn't have an atmosphere, there's no nitrogen on the moon. But
Right.
on Mars, they've tried to search for it with their rovers and their satellites. There hasn't been any proof yet, but there is speculation within the scientific community that's how the nitrogen could be formed.
Got it. All right, well, thank you very much. We are going to move on to Janelle.
Okay, can we all see the presentation?
Mhm.
Wonderful.
Are you ready?
Yes.
Okay, so it's my pleasure to introduce Janelle Ray, and her presentation is entitled Total and Mineralizable Soil Carbon as Affected by Tillage in the Argentinian Pampas.
Thank you, Chip, for that introduction. So real quick, I just want to tell you guys why I chose this. For the past year, I've been looking at how pre-plant fertilizer treatments affect total and mineralizable soil carbon within strawberry beds there at Cal Poly. So I wanted to choose a study that had a little bit of a similar sense to it. So first, an outline of what we're going to go through. I'm going to give you a little bit of background on the project and then the objectives, and then I'll go into the materials and methods, and then the results discussions, and then I'll conclude the presentation with a few takeaways from the study. So the site for this study was in the Argentinian Pampas, which, if you did not know, is just large rangeland over in Argentina. You can see on the map on your screen where the Pampas are based off of the little white oval. So this area is greater than 10 megahectares. Its soil is comprised mostly of mollisols and grass vegetation. It has a temperate and humid climate and is primarily used for agriculture. Now, because of its usage, this site over the past 100 years has shown that there's been a higher carbon loss relative to uncultivated soils than that of different areas. So one thing that has been proposed for this is to implement a no-tillage alternative to these seed beds in order to increase carbon in the topsoil. Now, no-till soils typically have a greater mineralizable potential, and past studies in Argentina have showed that no-till can increase carbon and nitrogen content compared to that of different tillage systems. And the southern cone of the Americas itself has a higher potential to be a carbon sink for a couple reasons. One of them being that previous intensive cropping practices have caused there to be a potential to store a considerable amount of carbon if there were improved management practices that were implemented. Another reason is that due to the small population that does inhabit this area, the agricultural pressure on natural resources is considerably low. So the two objectives for this study was to, one, determine the balance between carbon inputs and outputs in the soil plant system under two tillage systems, which would be our no tillage and our reduced tillage. And two, to establish whether these inputs and outputs could influence carbon sequestration. So now we'll go into our materials and methods. So this experiment took place at the Estación Experimental Agropecuaria Instituto Nacional de Tecnología Agropecuaria, which is located near Marcos Juarez, Cordoba, Argentina. This institute is used to research various agrotechnology practices while also looking at various agricultural research. It has an altitude of 107 meters above sea level and a temperate and subhumid climate with a mean annual temperature of 17 degrees Celsius, which is roughly 63 degrees Fahrenheit, and a mean annual precipitation of 924 millimeters. So this experiment was divided into 3 blocks, and each block was subdivided. The site was also implemented with a crop rotation of corn and wheat and soybean, and these crops were subjected to reduced tillage and to no tillage. Now, after 10 years, data was collected on the corn plots after they were harvested, and for each plot, 4 cores were taken, and also soil and stubble samples as well. For those of you who don't know, stubble is the short pieces of the corn crop that are still left over after harvest and it's been cut down. And this is also the crop residue that ends up being tilled back into the soils. Now, there were 4 different experiments done for this. The first was our total organic carbon, which was done through a wet oxidation. Our mineralizable carbon, which was done through an aerobic incubation. Now, this was done for a total of 10 days, and the soils were kept at near field capacity for water. And then the last 2 experiments were the bulk density and soil respiration. Now, bulk density was done through the simple oven-dried method, and soil respiration was done by having 6 cylinders, and within each cylinder, placing a flask with alkali in the soil inside of them and closing them off. And then after 5 days, the amount of alkali excess and carbon dioxide that was liberated from the soil was measured. So there are 2 ways to estimate carbon stock that they did. The first was sampling soils to certain depths. And this was done by estimating carbon based on soil mass and carbon concentrations obtained at that depth. And soil layers that were looked at were from 0 to 20 centimeters. And this was calculated, we used bulk density to look at that. And the carbon stock was estimated for each depth interval, and intervals were for 5 centimeters, so there were 4 intervals for each core. And data was analyzed using ANOVA and Tukey. Now into our results and discussion. So first I want to talk to you guys about total carbon. There was a reported estimate of 8.3 to 17.3 grams of carbon per kilogram of soil that was found. And through ANOVA and Tukey, we found that there was a statistically significant difference among sampling times and depth, and there was a statistically significant interaction between tillage and depth. That came from an accumulation of organic matter from the no-till and a more homogeneous distribution of organic matter under the reduced till. From this, we were shown that the total organic carbon was significantly greater in the no-till than the reduced till, only at 0 to 5 centimeters, so the first top 5. For potentially mineralizable carbon, this data reflected the labial carbon pools that accumulated because of carbon inputs and lack of decomp. Now, these results were similar to that of total organic carbon, where the carbon was greater in the no-till than that of the reduced till, again, in only the top 5 centimeters. Now, looking at our grain and total dry matter yields, we were shown that there was no difference in the amounts between reduced till and no-till at both the flowering and maturity stages. And residues also being put back into the soils were similar in amount and quantity between both of the tillage systems, and also root carbon content between tillage was equal as well. Now, soil respiration, CO2 respiration was significantly different between tillage throughout the entire 20 centimeters, showing that reduced tillage had a greater amount of CO2 emissions. And you can see in the graph on the right that both tillage increased the amount of respired carbon up until about 125 days and then started dropping from there. And for soil carbon stock, we found that bulk density was the greatest under no-till at the surface, and there was a highly significant interaction between depth and soil, and depth and tillage system, where no-till compacted topsoils much more than reduced till. Compaction produced greater carbon stock based off of depth, and both carbon stock and bulk density showed no difference between the 0 to 20 centimeters for all intervals. However, there was, for bulk density, it was denser at 0 to 5 and 5 to 10 centimeters for no-till than that of the reduced till. So just a couple takeaways from the study was that the total mineralizable carbon for that, the tillage systems didn't present any statistically significant differences, but a stratification was observed where as you went down in intervals, there was an increase of carbon that was found. For corn production, there was no difference between amounts produced between reduced till and no-till at both flowering and maturity stages. And soil under reduced tillage showed a greater amount of CO2 that was respired. Thank you. Dr. Appel, I believe you're on mute.
And so I was. So unfortunately, we don't have time for questions. Thanks for letting me know, Janelle. So we'll move on to Madeline. All right. So my pleasure to introduce Maddie Ropp and her presentation entitled Effects of Microplastic Biofilms on Nutrient Cycling in Simulated Freshwater Systems.
Thank you for that introduction. I would like to see a quick show of hands for how many of you like being outside. Looks like most of you. But when you are outside, how many of you have noticed plastic pollution? Unfortunately, that was a lot of you. And this is only the plastic you can see, but there's also microplastic pollution, which is just as harmful, and you might not even see it. Today, I will be going over the objectives, background, methods, materials, results, and discussions of the study. And right here, you can see an example of a freshwater ecosystem in which the study is focused on, and you can see the chemical composition of polypropylene, which I will be discussing shortly. So the objectives of this study were to compare the concentrations of nitrogen and phosphorus in systems that were exposed to microplastic biofilms and without microplastic biofilms. And this was to see if the addition of microplastic would alter the overall cycling in these elements. So plastic pollution is harmful due to its extensive use and mismanagement. Plastic is used almost every day and by many, many people. But unfortunately, this plastic is not properly recycled and it is not often handled correctly. So it often ends up in landfills and in pollution instead of being reused. Microplastics have been researched for quite a little while now, and a lot of researchers have made the term the plastosphere, which is when biofilms form on microplastics. These biofilms are really significant in many ecosystems, not just freshwater, because they're responsible for cycling many of the nutrients. These biofilms are composed of different microorganisms such as bacteria, algae, and extracellular polymetric substances. And when they eat and take in all these different things within the systems, they end up cycling the nutrients throughout the system, which allows aquatic systems to store many of these elements. So this research study really started with polypropylene, which is one of the most commonly used plastics and one of the highest sources of plastic pollution. And you can tell if the piece of plastic you're using is polypropylene if it has this number 5 recycling symbol. And this is so commonly polluted because it is not recyclable in SLO and in many other parts of the country. And right here we have polypropylene sheets in which the researchers cut into squares of 0.3 by 5 by 5 millimeters. And they actually put these into a pond at their research institute and allowed biofilms to grow within this system. After biofilms grew, they then collected these pieces of plastic and took them back to the lab for further study. They separated these into the treatment group and control groups. The treatment groups were broken up into T and T+, and T had 25 pieces of microplastic biofilms per liter of pond water, and T+ was the same, but it also had an addition of sodium nitrate. The control group was similarly broken up into C and C+, but there were no plastic biofilms in those. In order to find the total amounts of nitrogen and phosphorus within the systems, they used a calorimeter, which you can see right here. So in order to do this, you first have to dissolve the biofilms in sulfuric acid and hydrogen peroxide. And then you put it in one of these little sample tubes and put it in this machine, which is light sensitive. And it determines the concentrations by seeing how much of the light can be absorbed. This is really similar to the process of spectroscopy, which you may be familiar with. They also photographed the spatial distribution and decay over time of these biofilms using a confocal laser scanning microscope. You can see the principles of this right here, and to do so, they first stained the biofilms, and then when they put them in the microscope, they were able to use different wavelengths of light in the laser through the laser beams, and it was able to use fluorescence to visualize and photograph these biofilms. Here we have the results for the total nitrogen over time. And you can see here there are two main groupings. And up top, these were both the groups with the additions of sodium nitrate, and the two lower had no additions. So even though the two groups up top started with about the same amount of total nitrogen, over time, the group with microplastics ended up with more nitrogen, whereas the group without microplastics ended up with about the same as it started with. You see a similar trend in the other two groups down here. And for phosphorus over time, they started out with about the same level of phosphorus, and they all started to decrease over the first 5 days. This is because phosphorus is a really important nutrient for many of these microorganisms, and they take up a lot of them in the beginning to grow their biomass. After that, there's a split here, and the treatment groups started to increase and ended up with more phosphorus in the system than they started with. And on the other side, the control groups without microplastics ended up with less and about the same amount of phosphorus in the system. This is largely due to the fact that microplastics have large surface areas, so these microorganisms are able to grow really large biofilms. And because there's so many microplastics within a system, typically, they're able to grow many, many colonies. This higher amount of microorganisms allows a lot more cycling to occur, so a lot of these are cycled more frequently. Here you can see the biofilm over time, and square A is at day 5, square B is at day 15, and square C is at day 25. And the different colors represent the different types of microorganisms you can find. So the pink is algae, the green are the extracellular polymetric substances, and the blue, which is kind of hard to see, but you can see a little bit, is bacteria. So this shows that a lot of the colors are randomly dispersed within a slide, which shows that there's a fairly random distribution of microorganisms within a specific biofilm, and that towards the end of the experiment on day 25, there's significantly less organisms than there were on day 5. This is similar to the research for regular biofilms, as they also decay over time. Overall, there was a clear difference in cycling of nitrogen and phosphorus, as the groups with microplastics had more within their system. However, this experiment was in a controlled lab setting, so it did not show the implications this has on the natural environment. There needs to be more research to see how it the larger environments, especially as these freshwater ecosystems act as a sink for many of these nutrients. So when they're more out in the world, it could have potential effects. Any questions? All right, thanks.
Thank you, Maddie. So our next speaker is going to be Max Swinderman.
All right, can you see my screen?
You just see a blank. Now we can. Yeah.
We can? Okay.
You ready to go, Max?
Yep.
Okay. So it's my pleasure to introduce Max Swinderman, and the title of his research article was Using Aerial Photogrammetry to Monitor Glacier Dynamics.
Thank you, Dr. Fell, and hello, everyone. So yeah, I'll be talking about the usage of aerial photogrammetry to monitor glacier dynamics, in particular, debris-covered tropical glaciers in Peru. All right, so I want you guys to take a minute and look at this image, and I want you guys just to think about what the importance of this image could be or the context of it. And so this image is actually the oldest surviving aerial photograph we have to date, and it was the first one ever taken in America. And it was taken in 1860, and this is the city of Boston. And after seeing the potential of aerial photography from this photograph, the North actually used hot air balloons as recon balloons to track and monitor the South during Civil War. So I think it's pretty cool to see how simple photos like this led to development of aerial photography. And so for a brief overview, we'll be going over the background where this experiment was taking place, and the objectives or three main goals of this experiment, methods and materials, and the results and discussions, an overall conclusion and summary of their findings. So this experiment was conducted in the Cordillera Blanca mountain range in Peru. And the Peruvian Andes holds around 70% of the world's tropical ice, and 40% of Peru's glaciers are in this particular mountain range. And the glaciers are rapidly retreating due to global warming, as there's been a 25% reduction in glacier area from 1990 to 2010. And this is altering the downstream hydrology, changing the water supply, and affecting downstream communities as well as the overall ecology of the place. And there's fear for natural hazards such as flooding in areas surrounding. And the glacier lakes process has multiple factors such as the debris, geology, and topography of this area, since it is a debris-covered glacier. And we need more understanding of the dynamics behind this and what's causing these lakes to expand and also disappear over time. So the three main goals of this experiment were to successfully deploy a multi-rotor platform suitable. So that is a UAV or an unmanned aerial vehicle. And second is to measure glacier volume changes, velocities, and glacial lake changes. And they do that by generating a digital elevation model using programs such as ArcMap. And the third goal is to use these data sets provided to investigate patterns between these glaciers and understanding debris-covered glaciers more. So short list of materials that were used during this experiment were ground control points, and ground control points are is something you see like right here, and a UAV or unmanned aerial vehicle. And the aerial vehicle they used in the experiment, which I'll show in the next slide, is similar size to this. And computer programs such as ArcMap, Google Maps, and Agisoft to record flight paths and generating the digital elevation model. So methods or the steps taken in this experiment, the first step was constructing and designing the UAV and the ground control points. So this image on the left-hand side is a GNSS survey, which stands for Global Network Satellite System. And basically what this does is this can tell you the exact location, latitude and longitude of a certain area that you're studying, and that's where you put your ground control point, which is this right here. And the bottom right image is the UAV that they used in this experiment. And the UAV only weighed about 4 kilograms, and it could fly for up to 12 to 15 minutes at a time. And this picture right here is an example of ground control points and kind of explaining what ground control points do. So basically, what ground control points do is if you place them and scatter them in a large area, it can basically, over time, they have known coordinates because of the GNSS survey, and it can map out a large area if you have enough ground control points set up. So after doing that, they then set up aerial flight paths, and they did this using Google Earth to track the UAV and their flight path over the glacial tongue. So this is from 2014, and these are the flight paths, and the yellow markers are the ground control points. And in 2015, this is the separate aerial flight path they did, and you could see it differs a little as it got more narrow as they studied this band a little more than they studied a little more wider section in 2014. And in 2014, there were a total of 601 photos taken and 323 used. And in 2015, there were a total of 929 photos taken and 826 of those were used. And then this here is a kind of summary of what a digital elevation model generation looks like. So they use a program called Agisoft and a specific structure for motion algorithm to develop this. So it's basically taking your 2D photos that you take using the UAV and over time processing it, creating a 3D map to look at. And then the next steps after that were accuracy assessment and finding errors in computations and computing data from both years. So the first way they tested accuracy is checking the ground control point placement errors. So basically the ground control points where they were placed in real life and the differences in the coordinates that were found after making the digital elevation model. And then the error provided was in root mean square error, which is using the XYZ coordinates. And another way they tested how accurate the data was by comparing the GPS elevation to the elevation data they found in the model they made. And then computing data from both of the years, they did this through clipping the digital elevation models together. So if you took Intro to GIS, this is what clipping does. So basically, if this was a glacier boundary they wanted to assess, you put this over what you want to assess, and then this is the output. So basically puts it in the boundary area. And then what they also did was they place velocity effectors along where they saw continual ice melt. So these arrows are pointing in the directions of where they see the direction of ice meltage. And so for the results of this experiment, first talking about errors, in 2014 they had a much larger error range than 2015. And that's more of a result due to experimental errors. That's 2014 was the first time conducting the experiment. In 2015, they changed it by, you see these checkpoints. They put these checkpoints in minimal topographic variation. So there wasn't a lot of uneven topographic ridges or cracks or crests. So this made the digital elevation model for 2015 much more accurate than the digital elevation model for 2014. And if you want to compare the two, you can see slight changes in expansions of this larger lake right here. And then you can see how little ponds like this weren't there in 2014. And so for actual statistics, the mean elevation change was negative 0.75 meters, which actually resulted in 156,000 cubic meters of ice lost. And for surface velocity, they saw that velocities ranged from 27 to 4 meters per year. And they noticed that velocity was higher with increased slopes. So on the right section of the glacial tongue, the velocity was much higher than in the flat portion right here. And then the largest elevation change was 18 meters. In this photo right here, this is where the largest elevation change took place. And you could see, comparing this image in 2014 to 2015, a huge chunk of ice broke off. And this was actually 18 meters of ice that broke off. And besides the besides the upper part of the glaciers, there's also a lot of expansions and disappearance of ponds and lakes. And this is a closer picture of the ponds and lakes separate from the slope part of the glacial tongue. And as you can see closer, if you look down vertical as that compares the years from 2014 to 2015. Yeah, so debris played a large role in explaining why certain ponds were expanding and certain were disappearing. So the debris, as you can see in the picture right here, keeps ice under it insulated for a longer period of time. So chunks of glacier ice are buried under sediment, and that explains why connective paths between ponds and lakes were broken up from year 2014 to 2015. And continual ice melt over time, it's constantly changing the surface topography. And this, and there's also continual down wasting. An example was this calving face right here is 18 meters of ice could fall into areas like this, potentially expanding lakes to twice its size. And for conclusion, so this was the first time they ever deployed an unmanned aerial vehicle for this purpose, and they had to do it because of cloud coverage, as it's impossible to view glacial tongue like that. And we also found out that rapid ice loss was at exposed cliffs and water ponds, and we see how important of a role debris played in explaining why certain lakes were popping up and then certain lakes were disappearing, and explaining the overall trend. And overall, this illustrates the viability of UAVs and how cost effective and cheap it can be instead of sending real life pilots to areas like this. And overall, just looking back at the photo I showed of the first aerial photograph ever taken, it's pretty cool to look at an article like this explaining how they're able to create a digital elevation model, and how in hundreds of years we're able to use technology like this to really explore isolated areas in the world. And that's it. Thank you.
Up next, unfortunately, we don't have time for any questions, so we will move on to Jacob Timmons. But Max Niemeyer, will you be ready after Jacob's?
Yep.
Okay. So if you want, if your computer's running slow, you can email me your presentation as a backup, and I can present it off of mine too. So
Okay, I'll do that as well.
just as a backup. Okay. So let's move on to Jacob.
Can you guys see my screen?
Not yet.
Not yet. Let's see. There we go.
There we go.
Is that good?
That's great. All right, it's my pleasure to introduce Jacob Timmons, and his presentation is titled Wetland Soil Microplastics Are Natively Related to Vegetation Cover and Stem Density.
All right, thank you for that introduction, Dr. Appel. So as he said, today I'll be talking about wetland soil microplastics. So a quick outline of what I'm going to be going over. First, I'll cover the background of microplastics. Then I'll be talking about the objectives of this study. Then I'll move into the materials and methods, and then the results and discussion, and then I'll close it up with a little summary. Oh, so first, just before I start, can I get a raise of hands or a thumbs up for if anyone's heard of microplastics? All right, sweet. So a little bit of background on microplastics. Microplastics are plastic particles that are less than 5 millimeters in diameter, and they could be made from an array of polymers and chemical additives that exist in a broad range of colors and morphologies. Microplastics have been found in Asia, Europe, South America, North America, Australia, and Africa. They've also been found in freshwater and saltwater bodies around the world, and within the bodies of bivalves, macroinvertebrates, seabirds, and even fish destined for human consumption. So in the past, most studies on microplastics have focused on particles in the water column, beach sands, or in sediments with in like sparsely vegetated habitats. So the abundance and distribution of microplastics in wetlands are very poorly understood. So I think this study is important because it seeks to understand microplastics in a transitional ecosystem between a terrestrial and an aquatic environment. So on to the objectives. The main objective of the study was to investigate the spatial distribution of microplastics in a tidal freshwater wetland in Washington, D.C. And then they also wanted to understand how wetland vegetation and hydrodynamics influence the spatial distribution of microplastics. So on to the materials and methods. So the study the study was located in a tidal freshwater wetland in Kenilworth Park and Aquatic Gardens in Washington, D.C. And sampling occurred over a 3.75 hectare area of the restored wetland that was part of the park. So this wetland has a direct tidal connection to the Anacostia River in Washington, D.C., which is important because the Anacostia River is one of the top 10 most polluted rivers in the United States. So the study area included densely vegetated marsh, tidal channels, and non-vegetated wetland flats, which could be seen in the picture to the right. So in order to choose the sampling locations for this study, a 1 meter grid was overlain on a map of the study area, and then grid cells were identified as belonging to one of 4 habitats. The 4 habitats were mudflat, channel edge, dense vegetation, or driftline. And so for each habitat, 6 cells were randomly selected as sampling locations. So moving on to sample collection and handling. So in 1 meter plots at each sampling location, plant stems were counted, cover class was determined for each plant species using the midpoint of 6 separate cover classes. So after the plant stems were counted and the cover class was determined, a composite sample of the soil surface was taken by combining 5 subsamples for each plot using a metal cylinder that measured 4.8 centimeters in diameter and 5 centimeters in depth. So afterwards, each composite sample was mixed by hand, weighed and dried for 48 hours at 70 degrees Celsius. And then they were reweighed to determine soil moisture content. Afterwards, soil organic matter content was then determined by mass loss on ignition at 400 degrees Celsius for 16 hours. So moving on. So in order to separate and count the number of microplastics in each sample, the study used NOAA's marine debris program for sediments, which was modified to accommodate for like the elevated level of organic matter in the soil. So first off, each wet homogenized sample was passed through a series of sieves in order to retain particles ranging from 75 micrometers to 5 millimeters. So after wet sieving, samples were processed using wet peroxide oxidation, which was used to eliminate plant-derived cellulose, or otherwise known as like residual organic matter that was left behind. And then after this, the samples were dried and then scanned under a dissecting microscope at 40 times magnification. And then a metal forceps was used to collect all identifiable microplastics, regardless of the size. So then afterwards, the microplastics were then visually sorted into fibers and fragments, which could be seen in the picture to the right. So pictures A and C are examples of 2 microplastic fragments, and then pictures B and D are 2 examples of microplastic fibers. And then to assess the accuracy of the visual sorting under the microscope examination, the polymer identity of each microplastic was determined using Fourier transform infrared spectroscopy, or FTIR analysis. And so this graph is merely an identification chart of a subset of particles collected from each of the 4 habitats. So like, for example, polystyrene, more commonly known as styrofoam, was found in all 4 habitats. And then this could be seen by the little orange bar in each. So on to the results and discussion. I think the main takeaway from these results is that stem density and total cover increased significantly between the mudflat, channel edge, and dense vegetation habitats. And then this could be seen in the little red box. It's pretty self-explanatory, but I just wanted to outline that. And then on to microplastics. So the microplastics were found in every sample in all 4 habitats, which is pretty crazy to think about. And the average number of microplastics was 23,200 plus or minus 2,500 particles per square meter. So the main takeaway from this is that the number of total microplastics in dense vegetation was significantly lower than that found in other habitats, while the mass of microplastics was significantly higher in the channel edge habitat. So on to the results and discussion. So the main takeaway from the results was this, the microplastic number was negatively correlated with vegetation cover and stem density. So in other words, as vegetation cover and stem density rose, the number of microplastics went down. And so this supports the hypothesis that vegetation promotes microplastic settling from the water column and reduces their transport to the interior of dense vegetation stands. So in conclusion, here's a little quick summary. The study confirmed the presence of microplastics in this wetland of a highly urbanized river. Microplastic distribution varied spatially, and the total number of microplastics was negatively correlated with plant cover and stem density. And so I think the take-home message for my presentation is that vegetative wetlands are very likely to play a role in plastic cycling. They potentially acting as sinks, transformers, or even sources of microplastics. And in light of increasing releases of microplastics, I think the role of wetlands in plastic cycling deserves greater attention, and the application of constructed wetlands engineered for microplastic removal should be further explored. Thank you. Are there any questions?
Jacob, unfortunately, no time for questions. Gotta move forward, but good job. And Max, should we try it?
Okay. Can you see my screen or no?
Not yet.
Okay. Says it's sharing, but
You want me to use the one I got? Cue me.
Yeah, I think that's looking that way just because I can't really. Yeah.
Okay, take it away, Max.
Okay. So before I got cut off last time, I was saying that this is a study of an unconventional way to measure soil organic carbon, and it just happens to be in a degraded mountain landscape in the country of Lesotho. Okay, can you next slide, please? Okay, so let's get on with it. In this presentation about the journal article, I'm going to go over a few things we need to know about the background of the experiment, as well as the objectives, the methods and materials, the results and discussion, and finally, what can be concluded from this study. Next slide, please. Okay, first off, let's look at where this study was performed. Fieldwork was conducted along the Mokholatong River in eastern Lesotho, and that can be found in the bottom right map. That's the red dot is the country of Lesotho, situated in southern Africa. And the map on the top right is the country of Lesotho, with the study area being that little red rectangle. And the big map on the left is the study area, with each one of those little dots being a sampling site. And that river is that Mokotong River, as I mentioned before. It is an area of, oh, sorry, let me go back just this, sorry. It is an area of high endemic biodiversity, as well as being an area extensively used for small-scale agriculture. The experiment was a joint study performed by graduate students at 2 different universities. The 2 universities that participated in this are the University of Witwatersrand, Johannesburg, and the University of Limpopo, Sovenga. I probably butchered that, but I'm trying my best. Next slide, please. Okay, the topic of the study was to examine soil organic carbon using spectroscopy. This is possible due to the correlation between soil reflectance and soil organic content. Studies prior have found that increasing values of soil organic carbon are inversely proportional to an overall decrease in reflectance in the visible, near-infrared, and shortwave infrared regions of the electromagnetic spectrum. The final question we can ask about the background is, why is this relevant or important? Well, first off, soil organic carbon is crucial in its role in the carbon budget. For example, soil organic carbon is classified as the 3rd most important global carbon sink, and it contains almost double the carbon that is found in the atmosphere and the terrestrial living biomass combined. Soil organic carbon also contributes to stabilization of the soil and formation of aggregates, which can promote resistance to erosion. Another point of relevancy is that in agricultural systems, soil organic carbon depletion can lead to reduced crop yields, lowered moisture retention capacity, and reduced nutrient status. Next slide, please. So yeah, that's okay, sorry. Now that we have a little bit of the background about the experiment, we can examine the actual objectives of it. The first objective is to accurately estimate the soil organic carbon content using spectroscopy under both field and lab conditions. The second objective is to compare 2 different modeling approaches. The first being PLSR, or partial least square regression, and the second being RF, or the nonlinear random forest model. The third and final objective is to evaluate the role of spectral derivatives in determining outputs. The purpose of this analysis is to identify better modeling approaches for soil organic carbon using remotely sensed data, which will enable a quicker and more accurate evaluation of the soil organic carbon in poorly known areas such as mountains. Next slide, thank you. There are a few different tools and instruments used in this experiment. The most essential device used is the analytical spectral device, the ASD Field Spec 4 optical sensor, and that's that device on the top right there. This tool measures the reflectance properties of vegetation, soils, rocks, and bodies of water in the natural environment. It is a portable instrument able to be worn as a backpack accompanied by a field probe set. Its common applications are in atmospheric research, crop science, forestry, geologic mapping, ground truthing, and soil mineralogy. Along with the ASD device, soil sampling tools such as an auger and a spade were also used. To navigate to each sampling site in the field, a handheld GPS was utilized. ArcMap was also used to determine the locations of the sampling site, as well as develop a map of the site. Next slide. Thank you. The first task is to determine the sampling sites. These were generated randomly using Hoth's analysis tool within ArcMap. This produced 109 separate sampling sites to accurately represent the surrounding area. At each site in the field, 5 spectral measurements were taken at a height of 1 meter each. The final task done in the field was to take a surface, top 5 centimeters of soil, sample of about 400 grams for subsequent laboratory analysis. Excellent, thank you. As a part of the experiment, spectral measurements on the 109 soil samples collected in the field were made in the laboratory as well. All measurements were done on samples that had been air dried and lightly crushed in a pestle and mortar to a uniform fine sediment. Soils were scanned using the ASD device with the same exact methodology as in the field. The next step was to accurately measure the soil organic carbon content. This was done by the LOI, or loss on ignition method. Just really quick, I'll talk about that, the LOI method. A subsample, about 20 grams, was combusted in a muffle furnace for 8 hours at about 430 degrees Celsius. The loss on ignition was measured as the difference between the oven dry soil mass and the soil mass after combustion, divided by the oven dry soil mass. Once the LOI was completed, the next step would be to clean up the data and apply the two models. The data was cleaned up by removing noisy regions of spectroscopy readout. Okay, so now that we know the methodology used, we can begin to interpret the results of the experiment. Of the 109 soil samples collected, only 94 were analyzed for soil organic carbon content, because they had to exclude, I think they had a few outliers they had to get rid of there. This chart on the right side of the screen shows descriptive statistics of soil organic carbon in grams per 100 gram of soil, I believe. And it shows values with the whole data set, the 94, as well as the calibration and validation data sets. According to this is another one of those names, Kruskal-Wallis test for skewed distributions, there are no significant differences among the three data sets. Thus, we can conclude that both the calibration and validation data sets statistically represent the whole data set. Next slide, please. Okay. The two models used to fit soil organic carbon content to the spectral data were the PLSR and the RF. The results indicate that the PLSR models are more likely to overfit the calibration data set, causing some pretty chaotic looking graphs. If I could draw your attention to the right side, there's a spectral readout, and we have in blue the field spectral data, as in red the laboratory spectral data. And as you can see, they're pretty consistent, as you would expect, because they're essentially just the same soil, one under field conditions, one under lab conditions. So yeah, due to this fact, we can say that the nonlinear random forest, or RF model, is more accurate in this landscape. Another important thing to note is that important wavelengths to predict soil organic contents were also localized around the visible range at about 400 to 700 nanometers. Next slide, please. Okay, well, now we can look at the conclusion and wider implications of the study. The study conclusively shows that soil organic carbon values in degrading mountain landscapes can be predicted using different modeling approaches based on field and laboratory spectral measurements. Along with this, the best model was shown to be the nonlinear random forest model, or RF model. This study can provide insight for the local subsistence farmers by pointing out where there is an ideal soil organic carbon content for agriculture. The last implication is that this study can help quantify carbon storage within soils, and coupling this with land surface models of soil erosion can mean that carbon export from mountains can finally be accurately calculated. This is critical for carbon budgeting as well. As well as an evaluation of soil nutrient status. So, any questions, or do we have time for them?
Fortunately, no time for questions.
Okay.
All right, thanks, Max. So the next person is Emily Nash. I need to allow you to share the screen. All right, there you go. Okay, it's my pleasure to introduce Emily Nash, and her presentation is entitled Increased Dependence of Humans on Ecosystem Services and Biodiversity.
Thank you, Dr. Appel. So I have a question to start off, which is how many people here, by a show of hands, have traveled somewhere primarily for ecotourism? So hiking or going to a beach? Okay, so a lot of you. So that's actually one of the reasons why I chose this topic, and I'll be further going into that in the background of the research. And then I'll go over the methods of the research, the results, and then the conclusions that we've drawn from it. So the background is that ecosystems provide many services to us, and biodiversity is one of the important resources that ecosystems provide. The biodiversity gives us things like medicine, food, and jobs, and ecosystems provide services such as ecological sources, which is photosynthesis, the water cycle, things like that, cultural resources like ecotourism, which I talked about before, and aesthetic resources, recreation, things like that. Regulation, which is things like flood control and water purification, and that's actually where hydroelectric power comes into from river flow regulation and sediment retention. And then things like food and water also come from ecosystem services. So according to the Millennium Ecosystem Assessment, 60% of ecosystem services are being degraded or used unsustainably. From 1990 to 2005, we lost 3.1% of our total forest area, but we gained a 32% growth in GDP. This led the researchers to the questions of, are humans becoming more or less dependent on ecosystem services with economic development? So for this research, the researchers looked at 152 countries. 92 of these countries were hotspots, which were classified from the Conservation International Classification. And 81 of those hotspot countries are developing countries, while 11 of them are industrialized countries. They also looked at 60 non-hotspot countries, and they chose these countries based on data availability and chose them from areas all around the world, excluding Antarctica. So the three sources of information that they looked at in order to determine how economic growth was impacting ecosystem services was the roundwood production per capita, hydroelectric output per capita, and tourism investment per capita. And they chose these for various reasons. The first one they chose was roundwood production. They linked roundwood production with forest area and the ecosystem that it provided and the biodiversity that these forests gave to the organisms that lived there. They chose hydroelectric power because there's a direct correlation between hydroelectric power and total forest area in a country. And along with that, 80% of hydroelectric resources are actually in countries that are considered biodiversity hotspots. And the reason I chose this was tourism investment. So I personally have gone places with lots of nature. In order just to view it, I actually took this photo at Yosemite. And tourism has been linked directly with how much water area there is, coastline, rangelands, and mountain area. So this is directly related to the ecosystems there. So the results of this is that these three resources actually gave global economic benefit gains to the entire world of 32% from 1990 to 2005. And the countries that are hotspot areas actually had a growth of roundwood of 200%, hydroelectric power of 60%, and tourism investment of 200% over this time. And with this increase, it also had a positive GDP correlation, while non-hotspot countries did not have as much growth in comparison. So once this GDP growth happened, 71% of hotspot countries continue to develop on these three specific resources, while only 55% of non-hotspot countries continue to develop on these resources. So this shows that hotspot countries with lots of biodiversity actually rely more on ecosystem services compared to non-hotspot countries, and will further their development in these resources, which causes even more reliance on ecosystem services because of things like emissions. An example of that would be carbon emissions. So from this further development, in developing countries, they had an increase in 285.8% of carbon emissions. Compared to in industrialized countries, they had a 53.6% increase in carbon emissions. So this further relies on ecosystem services to do things such as carbon sequestration and to be a carbon sink. So the conclusion to this is that economic growth has increased human dependence on ecosystem services. So it's very clear that there's a correlation between economic growth and the reliance on ecosystem services. By further development, we even further rely on these resources in order to grow our GDP even further. And their second conclusion is that we're asking more from our ecosystems as our economy grows. So one of the key issues is that people often misunderstand conservation and think it's either too simple or too complicated. And we often view conservation as what resources we have now and how we can solve them now. But what we really have to think about is as we further rely on these ecosystem services, how we will have to protect them in the future, because it's not going to be the same amount that we're using them now if we continue to use them in the future. And so I want to leave you with, when you go outside next time, to really know that you're using these ecosystem resources and how you use them in your day-to-day life. Thank you.
Thank you, Emily. So we will move straight on to Caitlin, who is our final speaker for today.
All right. Can you guys see this?
Yep.
Awesome.
All right. So it's my pleasure to introduce Caitlin Thomas, and her presentation is entitled The Growth and Collapse of a Littoral Lava Dome During the 2018-2019 Eruption of Kato Bar Volcano in Papua New Guinea, Analyzed by Multi-Sensor Satellite Imagery.
Thank you for the introduction. So the first time I ever learned about volcanoes was in the 3rd grade, and I remember just being completely fascinated with them. And to this day, I still am. I think they're super cool. So obviously, I picked something about volcanoes. So without further ado, let's get into it. I have a brief overview that I'm going to be going into more detail about the background, the objectives, the methods and materials, the results and discussions, and lastly, the conclusion. So for the background, the Kato Bar volcano is a stratovolcano predominantly composed of andesite. It is, the summit is about 1.5 kilometers wide, and it has an elevation about 365 meters. And it has a very circular type shape for the whole island. It is one of 6 volcanoes that run perpendicular to the north of, or not perpendicular, my bad, that run parallel to the north of Papua New Guinea. And due to its remote location, as you can see on the image right here with the arrow pointing to it, it hasn't had much documentation for the eruptions. So the first eruption was recorded on January 5th, 2018, which was actually the eruption documented in this research paper. And there was speculation as to thinking there was an eruption before it in around the 1700s, but no one was sure about it. Going right on into the objectives, the objectives were to analyze, monitor the growth of the lava domes, and this is based on the multi-sensor satellite imagery. The satellite data allows the detailed monitoring and observations of the remote volcanic activity. Into methods and materials. There were many field photographs taken. As you can see, there's one displayed here on the left, and it was actually taken with an iPhone 5S. And I was quite surprised that it was taken with an iPhone 5S, especially since I learned that it was taken while they were passing a boat past the island. And so I thought it was a really great picture for just an iPhone. And as you can see, it points out the gas vent, the central cone, and those types of things. The field photographs were mainly used just to confirm the structures that were seen in the remote sensors. And then there was also, for the satellite sensors, there was optical, thermal, synthetic aperture radar, also known as SAR sensor. They mainly use the SAR due to it being independent of weather. So if there was cloud coverage, it would still record correctly, and also the small size of the island. The optical and thermal sensors weren't weather independent, so they weren't as precise of data. And as you can see on this lower image that's across the bottom of the screen, the red is the thermal activity, and it's in the pre-eruptive gas vent and where the arrow is, and that's at the center crater. And it, once again, since the exact positioning wasn't possible, they used it for more of a good idea for the layout and growth of the island overall. Right on into results and discussions. We have this picture right here. It shows, for the overall results, there was magmatic activity occurred at 2 main sites. It was the new peripheral lava dome that you can see at part B, and then there was the summit, as you can see on part C. And this shows the overall growth of the peripheral dome and summit craters. And actually the growth was about 3 times from the original. And so this goes from 21,600 square meters all the way to 665,500 square meters within about a month. So their average growth rate was 4,490 square meters a day, which is quite a lot. And then if we go over to B, you can see the growth over here, and its current growth, or like the original growth, is in the lighter colors, so the yellows, the greens, the red, and it had a growth rate of 380 square meters a day. And the red is the maximum growth rate, and right after the maximum growth rate, there was a collapse of it, and also the eastern side of the slope also collapsed of the island, and this caused a tsunami. So then there was regrowth, as you can see in the blues and purples. And then also for the summit, as you can see in C, there was a deepening of 2 craters. You can see once more of the north and once more of the eastern. And then this was created as more of like a simplified version of the last image. And this was the theoretical model of evolution and of the eruption, and it shows the activity for part A of the summit crater. And then part B shows the growth of the dome up into its maximum, which happened in February 7th. And then a couple days after, actually on February 9th, 2018, the dome collapsed along with the side of the slope. And then that released this tsunami, as you can see in the waves. And then a new dome was regrowed, and this was up until May 2018. And then after that, the summit crater emitted lava flows up until October 2018. And this began a new phase of the eruption, which was much stronger in activity than any of the other ones. And that lasted up until 2019. And as for the conclusion, this article covered the rare case of growing, collapse, and regrowing of the literal lava dome at Kavadar volcano, and the combination of the multiple sensors used made it a very historical and well-documented eruption. And I think as a takeaway, just the overall use of technology and how far it's come has been a really great thing. Are there any questions?
I have